we had an issue last time where if the connection to the projector ever gets interrupted, the recording stops. So on Tuesday we had our recording ended up in 2 parts, and I kind of struggled with that. And right after class, I went and I talked to people at the tech, classroom tech support about, the issue like that, and maybe they fixed it. But if you ever notice that you're sitting there and you're like, oh, the projector just went off and back on, and I don't seem to notice it, let me know, because it'll interrupt our recording like that. And so that's that. Okay, so here's this thing I was just talking about that I've talked about before in class. Ta-da. And then here's what we're going to do this week. Okay, so you got your exam back, and just don't feel terrible if you didn't do as well as you want, because I've taught this course a bunch, and no matter how I try to set it up, the first exam is just a really struggling thing, and then students kind of tend to figure things out and do a lot better. And I have a lot of respect for students that can increase their scores through the semester. I think that's what the purpose of college is to be kind of confronted with, here's a new instructor who, a new topic, wants you to learn it this way, right? And so you have to become a flexible learner, and, I think that's the big take-home, long-term value of college is just, learning about learning and learning about how to learn different things in different ways and stuff like that. And so, you and I can talk about it, and along the way, after the first exam, when students ask me, well, how can I do better? I'm like, so it's super like telegraphed from day one, and that is just you have to really learn everything in class. That's the first reason why I started recording stuff, because I'm just blabbing, and it's really deceptive like that. You're hearing it, aren't you learning it? No, learning is all about recall. There's no learning on hearing something. You have to recall it, right, like that. And I also think this is just a great topic for learning about the best ways that you learn. So, I'm super enthusiastic to be supportive and, try to help and talk about it. So, got to go over the exam. We actually have an assignment connected to it. It's not going to change your exam score or grade. It's just a new assignment, but it's forcing us to go back to it because the final exam covers everything. And then I think a big struggle in the first exam is just the format, the expectation of what what do we need to learn? And so, so we got to do that, and that's due on Tuesday. And then I posted the answer key video. I posted the grade distribution. I think that makes sense. I talked about those things at the start of class on Tuesday. Then I also just took some pictures of some anonymous student answers, because I think that's helpful to see. It's different to hear the instructor talking about this is how you should do it, and then see, oh, this is how a student did it, and it was a good answer, right? And so, I like to try to share some of those just to give you a feeling for, what the expectation is, what the target is for the answers like that, okay? So we're gonna be jumping into plants today. And so we have this flower assignment video, and we'll talk about that, in the class today a little bit. What is that about? But the flower assignment video is to watch the video, and it is self-explanatory, what you're supposed to do. And that assignment is due on Tuesday. I made the video, and I put this in the Bright Space post. I made the video 2 years ago, and 2 years ago we used it as a quiz. So it was a video to watch and be ready for a quiz. And I talk about that in the video. We're not doing the quiz, we're doing it as an assignment. So do it just like that, but instead of preparing for a quiz, you're writing it up like a take-home quiz assignment like that, okay? So that's where we're at on that. And to get there, we're just picking up a couple of topics that we would have liked to have covered. They're from chapter 4, so they're about animals and especially about vertebrates like that. And so we're just picking those up, and we're almost done with those, almost ready to go into plants. And these are topics that are just as important as any of the other topics we talked about. And we didn't get in, we kind of reviewed some and stuff like that, and we were pressed to get, all this stuff done. And so, we're picking them up here, but they're just as important as everything. So we talked about them already. We just need to talk more about this one. So how do you make a transgenic mice? And there are these two parts, right? There are ways to make good transgenic cells in a dish, and then there's a way to turn that cell into a mouse that will be breed true, that you could sell. And then some signal vectors sometimes function as morphogens, and we got a couple examples of that, and we'll see a couple more of those. And then the third one we have to talk a little bit about, more about, is this idea of a homeotic gene, okay? So I wanna, I just have some of those slides in case people have any questions about those. Does anybody have any questions about that or anything else right now? And we're doing great. Okay, so in making transgenic mice, I wanna just kind of go in and dive in on one of these examples, because you'll see it's really satisfying, where you're like, okay, I kind of get it that, right? Okay, so again, the story is only in mammals, and it's about the special embryonic stem cells in mammals. And these scientists won a Nobel Prize, and they really won the Nobel Prize just for that technology. It wasn't really a big new discovery, but it was a new tool that scientists could use, and that was a big deal. And this summarizes the whole thing. And the part of it is, well, we're going to manipulate the DNA in a test tube. We're going to get it back into cells, and that's really rare. So we have to use many, many cells and get lucky in a dish. And then we take that, identify that special cell, and we put it in a pre-implantation embryo. Then we put that into a surrogate mom, and we start producing these chimeric mice, and then we breed them, and we find mice that make the original special cell as sperm or egg. And as soon as we have that, we just breed them, and we maintain that line, okay? So there's an example of that here in the original video. It's a transgenic mouse that has had 3 different transgenes put in to glow green, red, and blue. And this is an example where those transgenes were put in one at a time. So you make the first transgenic mouse, we got one that glows red, like that, and you would take that mouse and we put another gene in, right? And those people at Jackson Labs do that, right? They make a humanized mouse with one, a knock-in substitution of the mouse liver enzyme gene with the human version. And they got that, they grow it up, they start selling it, but they take that and they make a second generation one that's got 2. And then 3, and it just becomes more and more of a humanized liver, which is a funny term, right? Because you're like, not going to go to school. The only difference is in its liver, and it's just in the subtle detail of how it metabolizes, how it breaks down certain drugs, okay? So we're focused in on that example, and we're just going to talk about the example of getting muscles glowing red, okay? Like that, okay? And so in this example, we have the first 2, the knockout, the knock-in, where we're replacing genes and replace the specific target gene by homologous recombination. And then we have the one we want to talk a little bit more about, and we're not going to change any of the mouse's genes. We're going to just add a new one. And the chromosomes are really long. There's plenty of spaces where there are no genes along the DNA of the chromosome. And if you just randomly insert a gene, it almost certainly will just be in like an unused space. So that's what's going to happen by non-homologous recombination. And so what we want to do is draw it out, okay? And so we're going to use this example of, hey, how are you going to make these muscles glow red? And to do that, we're going to go back to our original video, and we say, and from our first exam too, we say, okay, so we've got this gene, it's the MyoD gene, okay? And I mentioned that I just don't bother to write out, that's the transcription unit, that's the promoter, that's the enhancer, but you need to, because that's recall, and just keep recalling it and showing us that you know that and you're knowing it really well. So then here's the myogenin gene, right? So that's the next one in the pathway, activate these genes to make a muscle, right? And then here's a muscle gene like that, right? Okay? And so this is just straight out of the video, straight out of our first exam, and we got this situation where we're going to decide we're going to start making MyoD mRNA, and we're going to make MyoD protein. And that MyoD protein is going to bind here. And something we just want to pay attention to is anytime a cell makes a protein, it makes many, many tens and thousands of molecules of that protein. There's lots of those. And we're emphasizing this direct pathway, but almost always there are more target genes than just one. And so here's another target gene that MyoD activates. And there's plenty of molecules of MyoD. So the estrogen receptor, right, that probably has a few hundred target genes. There's plenty of receptor to be bound to everyone. There's plenty of MyoD to bound to everyone, okay? So we get that going, and it's gonna activate, it's gonna activate transcription of MyoD, and we're gonna get that, myogenin, we're gonna get myogenin going, and we're gonna get the myogenin protein going, plenty of myogenin protein to activate all the muscle genes, that are targets. It's gonna activate that, right? So that's that very familiar story, right? So what are we gonna do to make a gene that's going to be a transgenic gene that glows? Well, we want that gene to be active in muscles. And so we want to use a gene that's active in muscles, and we're going to use, in a test tube, the myogenin gene, and we're going to use that regulatory region of the myogenin gene in a test tube. That's the DNA we want. So that's part of our transgene, and it's the myogenin regulatory region. And I haven't drawn it, but because it's from a totally different species, and there is a jellyfish that has a gene that's called GFP, and that makes a protein, and the protein glows. And that has a gene organization like that. And from it, we want to get the transcription unit. So every gene is composed of two parts, and here we're just putting two parts together to make this transgene. It's a perfectly normal gene. We just put these two parts together. It's no problem. They're from two different species. We put together the regulatory region we want that's going to cause the gene to be expressed where, when, and how much, and the transcription unit, the product, right? And so this idea, right, should just be like, oh yeah, I get that, right? But like I was at a kind of a type of a job interview to work in a lab at MIT, and I was talking to the interviewer about this, and he was like going, oh my God, because his friend was trying to do this too, and he was like quietly, it's a long story, but it's a really funny story. It's because I was lying down on a couch. During a job interview, I was just like, I'm gonna just lie down on your couch here and relax and tell you stuff, yeah. But yeah, back in the day, nobody had thought about this. It's not that big a deal, but not that long ago. And that's what science research is like, is once you get up to speed, any of us could, yeah, we should do this. The challenge is do it, think of it. People will say, and it's surprising in science, this kind of science, ideas are like a dime a dozen. It's not the idea, it's the actual, well, now just get the work done, and the work done is not that hard, but it's kind of like thinking about a new recipe. I'm gonna make a new recipe and win the big contest of the new, baking contest and win $10,000. Well, you gotta do a bunch of practicing and, work out the details so it's really yummy like that. So it's surprising, but right, this should just be, for us, we should just be, yeah, I see what's happening there. Right? Because what's happening there is the transgenic animal will now have this transgene. And so here's the transgene. It's got literally a myogenin promoter and enhancer, and then it has this GFP transcription unit. And the mouse is totally normal because it got put in by non-homologous recombination, and you can see what's going on here is we have just created another additional MyoD target gene, right? Whenever the MyoD gets expressed, it activates myogenin. And there's plenty of myogenin to activate this transgene in exactly the same way, because it's exactly the same regulatory region, it's just another one. Okay? And so this transgene and the mouse, the transgenic mouse, the scientists that make that, they say this mouse is they call it a reporter It's a reporter mouse. Because you can study it, and it reports muscle differentiation. So in the video, right, in the early mouse embryo, in the video we start our class in, the early mouse video, it has all the genes, right? But it's not expressing these genes yet. And so there's no glow. And as soon as muscles start to form, just a little bit before the mouse's heart starts to beat, you see the mouse start to glow because it starts making muscle. So it's a reporter. And so it's really powerful tool for you if you're studying muscles, right? Because you could start with that transgenic mouse, and you could use that mouse in your genetic screen to look for mice that are mutants that don't have muscles. They don't glow like that, So, these people won a Nobel Prize because you're just like, oh my God. Once you see that, you're just like, oh my God, right? Because it's a reporter for muscle differentiation. If you're studying muscles, that's a cool mouse to study, right? It's also a reporter for myogenin activity. It's a myogenin activity reporter because every cell that has myogenin glows red, right? Because as soon as this natural wild type normal myogenin gene starts to get expressed, the transgene starts to get expressed at exactly the same time. This is also, it's also called a MyoD reporter gene, like that, right? Because as soon as MyoD gets made and does its job, the cells start to glow. So, you could be studying either muscle differentiation or the myogenin, role, or MyoD, right? And that's it. We just want to see that and be able to explain that on an exam. So, practice thinking about that and explaining it. That's the reason in the muscle differentiation videos I made the genes out of clay, because scientists just cut them up and mix and match them in a tube all day long. You can knock out, you knock in, you can make reporter genes, It's just driven, the technology, the concept has just driven this field forward so fast and so far, okay? Like that. Now, without drawing it out, let's just think, right? What would this look like if we wanted to study estrogen hormone? Well, we would do the same thing, but we would take the regulatory region of a gene that gets activated at puberty. We take that regulatory region knowing that the estrogen receptor binds to it, and then estrogen turns it on. And we connect that to the transcription unit from the jellyfish gene that will glow. And you would see a little kid running around or whatever, young vertebrate, baby fish, baby mouse, whatever, young vertebrate running around. They got all that stuff in them, they're normal, they got this extra transgene in them, and when they hit puberty, the animal starts to glow in lots of cells, right? Because the estrogen receptors get activated in lots of different cells. Not all of them, but there are lots of cells that are targets for change during puberty, like that, right? So, oh my gosh, you just go on and on and on, right? And that's just really satisfying to see that, to see, oh, you inherited all the genes, you got them, but they're not glowing yet because they're not activated. And as soon as they get activated at the right time, the right place in the organism, those cells start to glow, okay? So I wanted to go back on that and just, nail that down. And, I think at some point you will still probably, like before the final exam, you'll probably watch those muscle differentiation videos again because they're just central to the story in many, many ways. And the more you can recall that, the more it sinks in, the more you loop back to it, the more you'll go, oh, okay, I'm starting to get this, right? And get it by being able to explain it, draw it out, label it. Okay, so that's that. Just wanted to cover that and do it kind of separately, okay? So any questions about that or anything else? Okay, all right. So then just, I've got some of these slides we already talked about and, just in case people had questions. So we just got to talk a little bit more about this homeotic transformation, these homeotic genes, okay? So again, there are unusual genes that when mutated and have a hypomorph, a loss of function, instead of just losing something, you replace it. Homeotic genes have effects that are in space, in different places in the body. So you can have the wrong thing in the wrong place, okay? And there are a variety of different types of homeotic genes, and they just mean that. When mutated, they cause a homeotic transformation. And you'll see, we're gonna really focus on just one example of homeotic genes. And that's this homeotic gene that is a family of paralogs in all animals that have an anterior posterior axis, and they're called HOX genes, H-O-X, and they're transcription factors, and they set up the anterior posterior pattern. And a big example of that are somites. Somites are numbered just 1, 2, 3, 4, 5. And those are addresses, just like a street address. From anterior to posterior, every vertebrate has an address, like a house address. Yeah, okay? And so they're homeotic genes. And this just shows you a little map. So in the gene family in fruit flies, there are about 10 of them. And then in mice and most, I think, vertebrates, there are at least 40. And what's happened is just the entire family got duplicated twice at the very early step in vertebrate evolution. And so we go from, one family to 2 to 4. And that's unusual. Usually we just copy one gene over, but these get copied all together. Then the next thing to point out about it is that normally when there's a gene family over evolutionary time, there's different recombination and changes and so forth, and the gene family ends up just scattered on different chromosomes throughout the genome. There's no reason for them to be together. So it's very interesting to see that this HOX gene family is called the HOX gene complex, because they are all together, and they stay together over evolutionary time, and they're said to be collinear, and they're collinear with the body. So from one end of the family to the other, we go from anterior to posterior. So it's just a fascinating set of genes, and they're orthologs. So the colors match orthologs in the different species, like that. And they match up. They're always collinear. These at this end are orthologs, and they're for the anterior, and then the middle, and the posterior. And then you see there are not that many of them. There are 10 or 12 of them like that, And how can they give so much information about the address? And the explanation is they're said to be, and I think I have this on a slide. So this is a picture in the book trying to show this. And maybe it doesn't show perfectly, but the word is they work in combinations, or they're combinatorial. And that's a really simple idea, and it's this. House addresses are like this: 2, 3, 2, and 4, 6, 6, right? You can just take a few numbers, 9, 10, right, and make lots of addresses. And that's how these HOX genes work. They're expressed in combinations. And that's what it's just trying to show you here. If you express gene 1, you're at the address here. And if you express gene 2 and gene 1, you're at the next address. So it's combinations, literally like that. And the analogy I like is, you say you live at 322 East Main Street. And you come home and you go inside, and there's a family they're eating at your table. And you're like, whoops. You guys are in the wrong house. And they're like, well, no, you are, we live here. And you're like, well, I live at 322 Main Street. And they're like, well, this is 23 Main Street. And you go out in front, and one of the numbers has fallen off. It's in the same physical location along the street, but it's a different family's home. And that's what a homeotic mutation is like for this, is you get a change in this combination. And so instead of having, antenna in this address along the anterior posterior axis, it's like one of the genes got mutated, you lost that number, and you get a different address. And they're like, this is where the leg is, right? So instead of having, the antenna at that house, living at that address, it's now going to be where the leg lives. And it's just that simple. But you can kind of imagine, back in the day, whoosh, this is a big puzzle, right? Like, what's going on there? But it works out to be this kind of satisfying thing. And it's combination, and it's these proteins are called Hox proteins. They're all transcription factors, it's just a picture. You don't need to scramble to write it down. But this just shows they've been studied in great, great detail. So this is just the part of a Hox protein that binds to the DNA, so binds to the enhancer of target genes. And they're fascinating because they have lots of target genes, and they really are setting up the body plan like that. And so there's some famous examples where a mutation has caused a change. Like, for example, snakes have no legs. They have changes, mutations in their Hox genes, and that's part of the reason. A snake is kind of like the middle part of a body the whole way, something like that. You know, I think its ancestors had legs and they lost them, and especially in the front legs, they lost them because of homeotic transformation. And there are other examples of that, like that in vertebrates. So anyway, the Hox genes are not the only homeotic genes. They're just the most famous, but there are a variety of different homeotic genes like that. So these are called Hox genes, and they're called Hox genes just because they were the first ones discovered. And you know, here's an example where you can see there's a shift in position, because it's true for any animal that has an anterior posterior axis that we can understand that way. Okay, and so that these were really discovered in the first paper about them, where the people were like, oh my gosh, what is going on? The first paper that was published about them like that was in 1985, so 40 years ago. And what it was just like is the scientists were studying that antennapedia gene that causes the leg to come out of the head, and they, you know, nobody thought that that had any relevance for any other animal, because it's so weird. But when they were doing their research, though, one of the experiments they did is they just tested to see if it looked like other animals had a gene that was related, and they all did, you know. And so that was the paper. You know, they're like, we're studying this and in fruit flies, and you know, we look stupid, but you know, because who cares? But it turns out it looks like it's in every animal. And that was the first moment where scientists are like, oh my gosh, and maybe all animals have really similar development, you know, and they, you know, that they're just built with the same regulatory genes like this. And that, you know, and so 40 years ago really is when people said that's when embryology became developmental biology, because it's just like, oh my gosh, this is going to get pretty crazy pretty fast. And so just those guys. And so then here's a figure, and it's the artist for our book likes to make these really simple, you know, kind of schematic cartoon drawings. And I just want to show you this and talk to you about what's going on here. And what's going on here is it's a timeline, and you know, early and later, right? And this top is, the top row here is really the development of the neural tube. And the neural tube is going to be, this is going to be the anterior and posterior. And so over here you see the neural tube is become different. And you're aware of that. We have a forebrain, midbrain, hindbrain, spine. And that's true for vertebrates, is they have this organization of their central nervous system from anterior to posterior, like that. And and so that's what the artist is depicting here. And so early on, it is just a neural tube. The neural tube is just like a garden hose. It's equal all along the length of the garden hose. But we got to make some really significant, you know, differences in the anterior posterior axis. So the focus is on that. And the scientists are like, oh man, those Hox genes, I bet they really rock and roll in the neural tube. And the answer is they don't. Because, and we start to introduce this, one of the central concepts in early development is cells, and when they're precursors, like a myoblast, part of being a myoblast also is to express signaling factors that will help another cell get down its path. And it's really nice analogy back to you. You know, you have a fate, you're working on that fate, and you're talking to your friends about it, your neighbors, your friends, right? And you're talking back and forth. It's a back and forth conversation. And that's how we get complexity out of the deal, right? Is we take relatively simple setup, it's a lot of signaling factors, but if we have them talking back and forth to each other. And so here's who really patterns that midbrain, that forebrain, midbrain, hindbrain spine. And it's the notochord and the somites, so the nearby mesoderm. And that's where the Hox genes are playing a big role. That's where we get the thing of somite 1, somite 2, somite 3, right? So those are just somites, they're just getting going. But one of their early roles is to tell the nearby neural tube who you are. And you're like, I am somite 1, I am somite 2, I am somite 3, right? Like that. So the artist has depicted this in a really simple, kind of too simple way, but it's about cells talking to each other, and it's about the Hox genes, and the Hox genes patterning anterior to posterior, and the surprise that even though we have this very anterior-posterior organization of the neural tube, it's not within the neural tube particularly that Hox genes set up the address, like that. Yep, okay. And so here's the thing that developmental biologists have really focused on, and it's just a small little piece, and we're almost done with this, but just to show you this, to get a little feeling for how dramatic and how much this is all kind of orchestrated. So here's the forebrain, the midbrain, and then this whole stretch here is the hindbrain, and then this will be the spine, like that. And so see, they're carefully showing, that there's a lot of distinction along the anterior posterior. And a place where that distinction is really nice to study is this hindbrain in a very early embryo. And there are 8 of these positions or addresses along the length of the hindbrain, and they're called rhombomeres. So there are 8 rhombomeres of the hindbrain, and it's just showing you a little bit about how you might have learned, if you took human anatomy and physiology, about the 12 cranial nerves. And the 12 cranial nerves, many of them innervate in these different rhombomeres of the hindbrain. And they're just showing that we're going to use the somites and the position of the somites and the expression of the Hox gene addresses in the somites to communicate to even the details of the 8 rhombomeres of the hindbrain, to give them these different identities to develop the different roles of the hindbrain. And the hindbrain is the kind of the deep kind of animal brain. Like even if you're a vegetable in the hospital, you probably still have your hindbrain functions because it's your heart beating and swallowing and things like that, yeah? And so it's really essential that gets patterned out, organized, and who else can swallow? Every vertebrate. Who else is governing their heartbeat? Every vertebrate. So this is the hindbrain is some old school stuff like that, right? And so just a little bit of that, to look and see how kind of amazing this all gets put together. And it's just about the Hox genes. Just about those Hox genes, and they're just used as an example of homeotic genes. So you could have homeotic transformations if you have mutations in the Hox genes, and the homeotic mutations would cause changes in the rhombomeres. You might be having rhombomere 4 instead of rhombomere 8. So wrong address, wrong place, like that, okay? So that's that. I just want to cover that, and we're good with that. I think we got it covered, yep? So that's all stuff that really fits into that kind of first portion of our course, which is about early, especially vertebrate development. And so then we're going to talk about plants. And I think this is helpful, coming off the first exam. Part of this is, these are coming out as chunky pieces like that, where you should study these. You should do your assignments in different places, right? You should do all your assignments like in your living room for animals, because you like to spend time in your living room, and then that's where you think about them. And then for the plant stuff, we should just go and get in a closet and just be like, we're just going to be in here, and this is where we're going to learn plant stuff, really, because it's a famous way to learn a lot of stuff. Is to move your head around to different places, because this is just an isolated package, And we're just gonna talk really, we gotta read the chapter, and the chapter has a lot of pieces, right, like that. But they're really just 2 stories we wanna get out of it. And one story is the homework assignment, and it's about flowering. And then the other story is actually just comparing plants and animals, okay? Like that. And
Can
so,
I just ask a question before we
yes, we can do, yeah, anytime, yep.
So I just had a question about the Hox
Yeah.
genes. So you mentioned that something last class, but I just didn't really
Yep.
listen to what you mentioned. Why can't a human just like grow an arm on its head, like the fly?
Yeah,
There.
so this is the perfect slide to see that. So see, there's this redundancy. So in the fruit fly, here's let's see, make sure I see this. Here's Antennapedia. See, and we have orthologs of it. And Antennapedia, if you just mutate that, you're gonna get feet, the legs growing out of your head, right? But you see that here, we have 3 copies of it, right? And so it's not so easy to, we made a mutation and we saw the effect. Because there's some redundancy here. And this is where we could dive into a little bit about the history of life on our planet, because the understanding is that redundancy allowed for a kind of evolutionary opportunity. And so, that's the first thing, is we don't see that because the interpretation is there's this redundancy. We have multiple copies of it. But the multiple copies then also gave the opportunity to use this, and they say to deploy these genes in a new role. So what's called their ancestral role, and it's still current, but in evolution, scientists talk about an old role, the original role. The original role is anterior-posterior axis. These clusters are called A, B, C, D. And it turns out that D cluster was deployed in vertebrates that develop limbs. So the overall ancestral role of Hox genes in vertebrate limb development is to say, this is where we're gonna have these 4 limbs. This position is where we're gonna have 4 limbs, and this is the position we're gonna have hind limbs. And those are different, right? Arms versus legs, different address, right? Right? And we don't see a homeotic transformation where this person's got legs instead of arms because of the redundancy that's present in that ancestral role. It turns out that there's an additional, and so scientists say if something evolves a new role, it can continue to have its ancestral role, and then the new role is called a derived role. So that's evolutionary terms. The D cluster also plays this derived role where, we'll learn later in our course, there is a new body axis in a limb. This is called proximal, and that's distal. So in a fish with no limbs, we have anterior, posterior, dorsal, ventral, left, right. In a vertebrate with a limb, we have those axes and the limb axes, which are proximal to distal. And who's going to be, who's going to control that? And it turns out that the D cluster evolved to be expressed in a limb and help pattern the limb from proximal to distal. Yeah? So let me see now, there's something I want to not lose track of here. And so that's fascinating, right? And what's going on is genomic equivalence. Every cell has those genes, and what if we started expressing, what if we got mutations in the regulation of those genes, and we express them in a new place, a new time? They can evolve this derived role. So that happens, but then here's another thing. We see homeotic transformation in vertebrate limbs because there is so much redundancy. For example, my neighbor from my childhood, Margie Anfrenson, had a homeotic transformation in her arms, where her arms were not formed properly, and she was my neighbor, and we didn't know that. She was born with a birth defect in her arms like that, and but now when I look back and I think about Margie's arms, it was clear she had a homeotic transformation in her arms. So that's part of it, you know, right? A very interesting example is, well, we don't see it because they're redundant. But where they have this derived role, it's not all of them. They're not all doing it. It's just the decluster. And there, it's not so uncommon, right? But also remember that in general, you know, when there are big mutations in the body plan in mammals, they tend to get aborted. You know, they're not going to survive to birth. So you know, mom might not even know she's pregnant because they pass away so early in pregnancy, or you know, there's just a spontaneous abortion like that where the embryo is just not going to survive the childhood. Those can be examples of this too, because a lot of times the transformation is just, couldn't, survive, doesn't, make a viable organism like that. Yep. Other questions people have? Yeah, and again, this whole topic is just messed up. Just so cool. And to see that, and examples of that, and it's called Evo Devo, and it's just like, you want to talk about why there's diversity of life on Earth? Well, it's evolution. And how did evolution work? Well, it's like an origami folding plan in organisms that have all the genes in every cell. And if you start deploying these in different places, different time, who knows what's going to happen? It's not like it's going to happen the next generation, right? Because we know evolution takes tens of millions of years. But you can see, you get the big hint of, I see what plays out over evolutionary time, right? We get to this phylotypic stage, and then we just, see big differences in the diversity of vertebrates like that. Yep. All right, cool. Other questions people have? And before we jump into plants, your question?
This is all in like chapter 4 area?
Yeah.
Okay.
Oh, yeah, you don't need to look at it. So we never need to look at chapter 4. It's not going to help. You like that? Yeah, right? Just because, chapter 3 was a really long. Yeah, and I want to have you tackle, chapters, but not in a way where it's too much, right? Because chapter 4 is a long, big chapter, and it's got a lot of because it's kind of, jumps forward in, like, let's look at more complicated stuff, right? And we really want to get these principles so that, you can hang on to them and, in the future like that, and, and not get overwhelmed. Yeah, so it won't help you. I'm not going to ask any, questions that would help to have looked at chapter 4. Like that? Yeah. So then before we play into plants, let me just mention sort of the flow of our course. So here we have plants, and it's just going to be an isolated package. We're going to talk about this chapter, and we will not talk about plants again in our course. Then, after we finish plants, we're back to chapter 2, it's fruit flies. And in that chapter, it's a giant chapter. We didn't cover it yet, because if we went from chapter 1 to chapter 2, all of you would have dropped. You would have said, this is before the drop date, I'm out of here. Because we have a huge chapter about freaking stupid fruit flies. What a useless waste of course this is, right? Like that. And so for fruit flies, it's a giant chapter because of orthologs. And the scientists were just, in the 80s, Antennapedia, they were just jumping on the bandwagon, And, oh my gosh, yeah. And so they discovered them all in fruit flies, sonic hedgehog and so forth. That's why they have dumb names, is because fruit fly geneticists pick dumb names for genes like that, yeah? And but in chapter 2, what's going to happen is we're going to say there are 3 chunky stories from fruit flies that we want to get out of it, and that's just a better way to tackle that chapter. So we're going to tackle some chapters, but not 4. We're going to tackle plants, that chapter plants, then fruit flies, and then we're going to go back to chapter 5, 6, and so forth, and those are back to animals, and we pick up kind of where we left off. So we're leaving these off, but we'll pick these up after this little foray into plants and fruit flies. Yeah. All right. Other questions people have? So plants. So the flowering video is about this species, and it's a weed. It's a little tiny weed. It's not in North America. It's in like Europe and Russia, sort of northern climate, like Maine, but not in Maine. And you just find it like growing in like little rock walls and cracks in sidewalks. It's just this little tiny flowering plant, and it only grows about this big. like that. And it has some common names, but they are a bunch because it's, you know, it's not a thing. You'd be like, oh, those are tulips. It's got lots of little funny little common names. So people just call it Arabidopsis. So the genus is Arabidopsis thaliana, and people just call it Arabidopsis. So it's Arabidopsis, and it's a flowering plant, and it's the fruit fly of plants. They were just way ahead. And one of the things that's an advantage in this species, it has a pretty small genome. So you could study it pretty fast. It's a pretty simple little plant. If you said, you know, who cares? People would say, all flowering plants are the same. You know, when we talk about animals, we are talking about wind and wind signaling pathway. When I say there's a wind signaling pathway orthologs in every animal. So that's true. Every plant, every flowering plant, anyway, has orthologs, just set up the same way. Every flower you'll ever see, no matter how diverse it looks, it's built on the same set of ortholog setting up the plant, like that. And so people would just say, it doesn't matter, you know, it doesn't matter that this isn't food you'd eat or, you know, like that, right? It's just, you know, it's a model organism, and you can grow it really fast like that, okay? So then the next thing they would mention is it's in, and I don't know much about plants, but it's in the mustard family, and there's a set of vegetables that we eat that are all related to mustard. And I don't know if you knew this, but like Brussels sprouts, broccoli, cauliflower, are all really, really closely related. And they just became different by selective breeding by farmers over time. And they're in this mustard family. So in that sense, this is kind of a good example for certain types of vegetable crops like that. But it's not limited to that. You know, it's everything like that. And so this is, we'll talk about Arabidopsis, and we'll talk about it making flowers. And just about all of that is in the video. So I'm trying to make that encapsulated and explaining it. And what is especially valuable for us is how Hox genes pattern the flower. So as this is growing, it's growing, and it's just growing leaves and stems. And I should write this down. In plants, that's called vegetative growth. And then later, it will switch to flowering growth. So vegetative growth is when you're just making a plant, stems and leaves and branches and so forth. And that's vegetative growth. And then when it's time to flower, that means reproduce. So then it's going to switch to reproductive growth. Like that. And so there are the buds on the tip of a plant, and the buds are growing, and they're making new stems and new leaves, and then the bud is going to make a flower, like that. And that's this transition. And if you look at, like, down at the bud, it's going to make a flower. It turns out all flowers have Hox gene addresses that look like this, like a target, like the bullseye. And they're just 1, 2, 3, 4 circles, and those are called whorls. And that from outside to inside, the first whorl is called the sepals, or sepal, and those are like the green covering of the bud. So the bud is forming, it looks green, and there's going to be a flower coming out of that, and the overarching, they're a little bit like leaves, and they're covering the bud on the outside. That's going to make a flower. Then the next one are the petals, and the next one are the male parts, and there are some different terms that botanists use, but developmental biologists call the male part the stamen. That's going to make pollen. And then the central part is called the carpel. And again, there are some different terms that botanists use, but those are the terms for the reproductive parts. And see, they're in, like, the carpel is the bullseye, right? Like that. And So there are homeotic transformations. So we could have a homeotic transformation that doesn't have any petals, and maybe it has more stamen. And so it's a feature in this Arabidopsis, and then orthologs of those genes, those homeotic genes, are found in every plant that makes flowers. So that's a big focus of the assignment that's due on Tuesday, and that's why it's called the flowering assignment, is how you make a flower. And of course, this plant has a life cycle, and it's vegetative and then reproductive. And we're not going to really talk much about the other stages, so like germination and forming the seedling and stuff like that. Big focus is really on vegetative growth transforming into that reproductive growth. And this is a good example of that. Okay? So that almost all of the first package of what we want to get out of the plant chapter is the video and learning about this Arabidopsis as a model of flowering plants. So then the second story is going to be about like a compare and contrast between plants and animals. So compare and contrast, that'll easily be a question on the second exam or the final, is compare and contrast plants and animals. Because there are only three kinds of multicellular organisms on Earth. There are plants, animals, and fungi. And fungi are come out, kind of branch out, come like from the animal branch. So people kind of say, really, we're talking about plants and animals. And the significance is that in making a comparison and contrast, because all the animals have really similar development. They all have wind and frizzled and so forth. And there's no plant that has wind or frizzled. But all the plants have genes that are orthologs, like these flowering genes, you know, like that. None of the animals have those. And the explanation, the interpretation is the last common ancestor between us and plants was a single-celled eukaryote. So it didn't develop. So this developmental biology evolved independently in plants and animals. So it's going to be different genes. It's a big explanation, you know. The molecular explanation for life on Earth is just like, that's what's, you know, that, you know. If you're an animal, you know, you know, evolved the developmental processes with animals. So you have a lot of similarity, a lot of orthologs. If you're a plant, well, you're going to have a lot of orthologs with other plants, but not in between them, because the last common ancestor was not multicellular, it was single-celled. So any evolution of multicellular is. And as a result, comparison of plants and animals on Earth for developmental biologists is fascinating. Because, for example, we just mentioned in this class, animals have homeotic genes. And they provide spatial addresses in a combinatorial manner. Well, it turns out plants have homeotic genes. And they provide spatial addresses in a combinatorial manner. And it's just like, right? Because that evolved independently. Are there Hox genes in plants? No. Nope. Right? The homeotic genes evolved kind of a similar role, but they're not related. It's independent evolution. And so that helps us see things like, I guess that just the concept or principle of homeotic genes must be a really useful thing. You know, if you could evolve that, you could probably make a lot of different plants. And if you could evolve that, you could make a lot of different animals. So see that comparison, that compare, contrast, what's similar, what's different, it gives us some really interesting insight, you know, because if we just look at animals, well, they're all kind of the same. But plants are really different. So how are they different and how are they similar? And that's all we're going to do in this second chunk. And the second chunk is just scattered throughout the chapter 12. The authors don't pull it together, but we're going to pull it together in class and, you know, kind of create an essay answer of how are plants and animals similar, how are they different in development, okay? Like that, okay? So here's just a evolutionary tree. You don't need to pay close attention to it, but common ancestor of all eukaryotes, like that. And so there that is, and then they were single cell, and then we get fungi and all animals out of here, and we get all the plants out of here, like that. And then other simple single cell eukaryotes over there, like that. So that's it, just, you know, development evolved independently in plants and animals, okay? So we're just gonna talk through, oh, and I got this, it says chapter 13 on this, that's from the previous edition, but chapter 12, comparing plants and animals, okay? And they invented or evolved multicellularly independently. And so we're just gonna have a short list, and it's not in the book, it's just a list that by kind of pulling things together. If you said, how are they similar, how are they different? We'd say, well, here's some similarities, here's some differences, right? So we're just gonna kind of memorize this list, and we gotta talk about it, because some of this stuff is new, right? Like that, talk about them a little bit more and show some examples. So here's some differences, right? 1, 2, 3. So let's focus on 3 differences. And we're not surprised, they're very different, right? They're very different. Plants and animals are very different, and we're not surprised to see differences. So here's the difference, and it's something we haven't talked about yet in animals. And it's just this line. There are no germ-line cells in plants. Okay, so what's a germ-line cell? All right, so we get to go over here and talk about this, because we're going to explain it first in animals. All animals have a germline, and plants don't. So what the heck's a germline? So in talking about plants, we just have to talk about animals and plants, and sometimes we have to add something new. So we're going to add something new. So the germline, okay? And so germline are in all animals. And all animals have these germline cells. And they are cells that can make the sperm or eggs. So the germline cells are reproductive cells. There's I was remembering there's a quote from 150 years ago that developmental biologists say, and I was just looking back into it, like, who said that first? And the person who said it, this quote, is actually like a novelist. They weren't a scientist at all. But the quote is, a hen, so a chicken, a hen is just an egg's way of making another egg. Like that. And that's what this is about. They didn't really understand that then. The author was using that for a different kind of analogy, but it's perfect in developmental biology. Our bodies are just suitcases carrying around our germline cells. Those are the only cells that matter. Our bodies are mortal. The germline cells have so far been immortal, right? So your parents' germline cells led to you, and your grandparents' germline cells led to them, like that. So the germline cells are really special. We find them in the gonads, the testes or ovary. Not all the cells in the testes and ovary are germline cells. It's just some of them, and it's just the cells that can make sperm or egg. In us, in the testes and the ovaries, there are some stem cells that make the sperm and eggs. And those stem cells are the germline cells. So all animals have that strategy. And then, let's change the color here, because the other cells, so the rest of our body, including the rest of our testes, ovaries, and our entire rest of our body, brain, heart, and so forth, those are called somatic cells. Soma means body, so they're somatic cells. And that's just the strategy. The strategy is to have a body of somatic cells carrying around the germline cells in every animal. And every animal is similar. That's the strategy, okay? And we'll talk about, now that we've introduced that, we'll talk about it some more along our way, like that. Because there's just, the germline cells are fascinating. Here's just a sidelight about how fascinating are the germline cells. Here's a fascinating fact of the germline cells. Remember that we learned that when our moms were pregnant with us, our bodies were made by us. So our cells made our embryonic, fetal, and baby bodies. And those embryonic cells also contributed to the placenta, the amniotic sac, and other extra embryonic tissues. So if you look at a placenta, the placenta is some cells from mom, some cells from the baby. And they don't get combined, they don't fuse together, they're separate, but they collaborate to make the placenta, okay? So here's how messed up the germline cells are. The special cells that are the only cells that matter start out in human and other mammalian development. They start out, and they're outside of the embryo. They start out as extra embryonic cells, and then they crawl into the embryo. And in vertebrates, it's true always. And how they crawl in is different. In birds, they get into the blood vessels and are carried through the bloodstream to the gonads, developing ovarian testes. In mammals, they crawl in through the digestive tube, through the archenteron, and crawl up there, And you're just like, those are the only, you would think that those would be the whole story of development, because those are the only cells that matter. And when you look at it, you're like, what's going on there? Yeah, it's just really interesting. And because of that way they developed, they were really hard to study until scientists were able to make transgenic organisms in which the germline cells would glow. Like that, right? Because when they could glow, then you could track them and be like, look at the glowing cells from these birds are going into the blood, and the blood cell, the bloodstream has got glowing cells in it. Look at them going into the mammal, it's the digestive tube that's starting to have cells in it glowing. So super crazy stuff, and very interesting. That's animals, okay? So we gotta cover that and learn about that to say there's no such thing as a germline in any plant. And we were just talking about that. So we talked about how the buds make the vegetative plant, the body of the tree. And then those buds change. They were stem cells, busy making leaves and stems and branches and stuff, roots. And then they change, and the buds above the ground, those can start to make flowers. And the flower is the reproductive part. The flower has the equivalent of the reproductive cells, right? It can make sperm and egg for the plant to reproduce. So that's, you just be like, that's crazy different, but I'm not surprised. That's just a classic example where plants and animals are dramatically different. Plants, all the same. Animals, all the same and different. So it's a great example. People like to say plants are easy to clone, and that just means you could take plant cells and just make more plants out of them. They seem to be just more ready to do that. Both plants and so it seems to be that they the cells maintain a sort of more of a flexibility than animals do like that. But then also, because of this, there's no such concept of a maternal factor in a plant. Mom is not helping to set up the embryo at all because of the very different strategy. Mom doesn't have these germline cells. Mom is just going to plant. The plant is just going to change. These cells are where they were making the body. Now they're going to make the flower, including the structure of the flower and the reproductive cells in the stamen and carpel. So that's the first one. Just no germline cells in plants, and that's very different than animals. So then the next one is no morphogenesis in plants. And so remember, in broadly, morphogenesis means the creation of form. So in that sense, plants should have, you should agree, they have morphogenesis. But developmental biologists don't use morphogenesis in just the overall generation of form. They really mean cells moving around. Because we say morphogenesis starts at gastrulation. That's when the bell gonna go loop bending in. That's when, you know, the cells are gonna start moving around like that to help make the body. And as a result, fate maps in animals are pretty interesting because you map a cell here and it's gonna move around. But you know probably already, plant cells have cell walls. They're not moving around at all. So there's no morphogenesis. The only thing that's happening is the cells are just dividing. You know, if you look and you like, you know, mark a plant, make a fate map like that, you know, like a little seedling, you know, littlest seedling, and you say, I'll fate map of this. Well, those cells are gonna be roots, these cells are gonna be leaves. You know, the fate maps are just boring because they're just where they're gonna stay. They're not gonna move around at all, like that. And so they have these cell walls. So it's kind of pointless to make fate maps because you're like, well, I know what that's gonna be. It's at the bottom of the plant, it's gonna be roots, like that. So it just lays out like that, all really straightforward like that, because they can't move around, can't crawl around, can't be pushed around. And so that's the second one, you know, and it's just very different. And you'll see that we'll want to talk in, we'll go back over this and we'll talk into those examples a little bit, you know. So what's, you know, what's the big deal in plants? The big deal is this, just to mention it, and that is, you know, here's mitosis, right? This cell is going to divide in two. Oh, it divided in two, like that. And you learned that in intro bio. It's going to divide in two. Oh, look, it divided in two. Well, it turns out that it's different if it goes like that. You know, it's the same deal, but what is the orientation relative to the rest of the organism? And that's how plants create form. It's just the orientation of cellular division. You know, and that, it turns out that matters some in animals too, but it's just really foreign and weird for an animal like us to look at a plant and go, oh God, your cells aren't moving around at all, and you're just going this, like that. Yeah, so fascinating, and we'll dive into it a little bit because it continues to be interesting to compare and contrast it. Then here's the next one, right? We have three differences, and they're very different, and we're not surprised like that, okay? The third difference is that plant cells contain these little donut-shaped tunnels called plasmodesmata. And there's a broader term for it, and that broader term is the gap junction. So animals have gap junctions, plants have gap junctions. It's a cellular thing. In the cell membrane, there are these donuts that can connect the plants together like that, or the animal cells together. But in plants, they're very big, very, very important. And so let's just draw that out. We're doing great. So let's just draw that out. So again, you know, it's compare and contrast. So we look at animals and we learned that there are source cells and target cells. And we can have a signaling factor, you know, come out of the source cell and affect a target cell. And these are in animals. And we kind of are aware that, you know, well, if that signaling factor is Wnt is a protein. So back here, there's a Wnt gene, and it has to get made. We have to go make that Wnt protein. And so back here in the source cell, there must be a transcription factor that's activating that. And then we know Wnt binds the frizzled, and you know we get that signal transduction, disheveled, and JSK and stuff like that. And ultimately, we stabilize beta-catenin, and beta-catenin is a transcription factor that's going to activate, overcome repression of TCF and activate some target genes. So just in looking at this, you could say, I can kind of see how this purple transcription factor can affect gene expression in that other cell. Transcription factors stay inside the cell, so how do they do that? And the answer is they do it indirectly. This transcription factor activates a signaling factor that leaves the cell, and then that signaling factor binds the receptor, and that signal transduction factor affects transcription factor, other transcription factors that regulate gene expression. So we didn't talk about that, but we understand that. So in plants, there are these plasmodesmata, and what that means is, I'll draw several plant cells, And they're right, these are just their neighbors, right next to each other like that, they're all like that. And plasmodesmata are like donuts or little tubes. And they connect these plant cells, and there's like a little tunnel. So those are plasmodesmata gap junctions. So in a plant, we could just have that purple transcription factor. It could just go float right through there. And we could have that purple transcription factor end up way over here. And affect gene expression more directly. Nothing has left the cell. So in plants, we say they're plasmodesmata, and as a result, there are very few examples of signaling factors in plants. We don't really have to have some source cell make a signaling factor that leaves the cell, because all the cells are connected. You've heard of probably xylem and phloem. Xylem and phloem are really just dramatically specialized examples of cells connected by plasmodesmata. That sap is going through plasmodesmata. Just connecting and going all the way up and down the tree, like that. No? Okay? And so that's our third one. And we're doing great, and we got to stop. And so you'll see, next Tuesday, we'll switch it over and we'll talk about some differences, but we'll also, fill in some details like that. But it looks like we got 5 similarities, rather, we'll talk about. So we'll make this compare and contrast. And we'll also use that to kind of dive in and learn a little bit more about plasmodesmata and so forth, okay? So we're all set for that. And remember, you have 2 assignments.